Next up, we have Fivos Perakis, who's from Stockholm University. And he's going to tell us about solution scattering and also X-ray photon correlation spectroscopy, and some very recent work that I think they published this year, actually. If you want to take over and go ahead and take it away, Fivos.
Thanks, Rich. It would be nice if we could meet everyone in person. Thanks anyway for the opportunity to give a presentation here. It's been very exciting to follow this new developments. I'd like to start by showing an image of Stockholm. That's not exactly how it looks like, but it will in a few months when the snow melts. So if you happen to be in town, please send me an email. I'll be happy to meet up and show you the lab and arrange a presentation for you. So with that, I will tell you a bit about our new work at the European XPCS. It's actually not published yet. We're working on it. So it's a lot of really new stuff I'm going to talk about today. And I'm going to start with this nice slide that I'm using in a lot of my lectures. My son likes that a lot, who is 2 years old, because it has a lot of ducks in there. So you see here on the left a nice lattice in real space, a square lattice, which would correspond to a square lattice in reciprocal space. Now, when you introduce a small amount of disorder and you displace randomly a little bit these ducks around, what you see is that on the right manifests a kind of diffuse scattering, a speckle pattern. And this is going to be now the focus of this presentation. I'm going to talk about how by following this speckle pattern in solution, we can get information about the dynamics. So this is now, in a nutshell, basically my presentation. We are using hard X-rays at the European XPCS, looking at the protein solutions. And we have a sample environment. And then we have the detector. This is a 2D array detector. And by following basically at one particular Q or one particular pixel, the intensity as a function of time, you can see basically these speckle patterns. They start to fluctuate. And that fluctuation here, this intensity fluctuation, has to do with the fluctuation that is happening in real space at the particular length scale that you're looking at, the particular Q. So by calculating essentially a correlation function as a function of these intensity fluctuations, that's shown here on the right. It's this correlation function G2 as a function of time. You can get information about the time constant. Very often we refer to the intercept as speckle contrast, and we plot this as log time. So that's how an exponential would look like, for example, here. And this technique is called X-ray photon correlation spectroscopy. So mathematically, what we have is this kind of correlation functions that you essentially correlate the intensity at one Q at one time with the intensity at the same Q at a later time. And this is now averaged over different, triangular brackets indicate averaging over different pixels. And there's a normalization factor that takes into account intensity fluctuations. So basically, by looking at the changes in the speckle pattern, you can get information about the dynamics, like is shown here on the right. And this is now some estimations we did for protein solutions, assuming a certain Q, a certain momentum transfer, shown here on the upper right. And you can see this is now for different concentrations, because depending on the concentration, you also change the viscosity. So these are for very high concentrations. This is what we like to call crowded environment, where things are starting to become very interesting because it deviates from what you would expect in a dilute case. So basically, the concept is like this. You look at the correlation function and you start with very early times, you have a very high correlation. And as the molecules are moving away from their initial position, you start to have a loss of correlation. And then, but depending on the Q that you're looking at, you're looking also at different distances. So 2 pi over a Q factor comes in there. Another interesting feature of XPCS, X-ray photon correlation spectroscopy, is that one can look at the heterogeneous dynamics. So what I explained now is how you can just get a time scale. But you can also calculate what I like to call here as a 2D XPCS. So instead of having a one-time constant, you introduce a second time constant, a T1 and a T2. and you calculate basically two correlation functions, or let's say correlation function as with two times there, and you get a 2D map that looks like this. So red means high correlation, and blue means low. And basically, by taking cuts along this axis here, you can recover the 1D correlation. So if your system, like in this case, is like I showed here, is equilibrium, so nothing is changing while you're measuring it. It doesn't matter whether you take a cut at an early time or at a later time or at an even later time, like shown here. You will always get the same kind of correlation time. Mathematically, this is now the 1D case that I showed you. The 2D essentially is a correlation function that includes I of q t1 and I of q t2. And here is now averaged over different pixels. So now let me show you some examples of how this 2D XPCS can get a bit more interesting. This is the equilibrium example where I explained that it doesn't matter at which point we take a cut, it will always have the same behavior, your system. It's always the same time scales. But you can also have a system that looks like this, where things are fluctuating as a function of time. So if you were to average over time and get the time constant out, it would be the average time constant, but you wouldn't have access to the fluctuations, to how much does this the time constant changes during the measurement. This can be due to some heterogeneity in the structure and dynamics, could be different reasons. Another aspect is it could be that something happens in your measure during the measurement. So initially you have something that is very narrow, very fast dynamics, and suddenly it blows up. It becomes very slow. It could be like a phase transition or something that the beam does to your sample, many things. But this is now just a demonstration of if you were to average over time and get the correlation function from these three cases, they would be identical. You would not be able to distinguish. But by looking at the 2D maps, you can get higher order information. And now to make it a little bit more relevant, here now some of the XPCS experiments that were performed at MIT, at the MIT station at the European XFEL, from our colleagues, Felim Lemkuller and co-workers, which basically they use this interesting pulse structure of the European XFEL. This is one of those things that you either love it or hate it. I know a lot of people have been discussing about this pulse structure that in this case, I think it's a good thing. So you have basically a lot of X-ray pulses that come. We call it a train, and these are separated, can be down to 220 nanoseconds. And then you can have up to 200 pulses or so here. And then there's a gap, and then there's another train. And these trains are coming at a repetition rate of 10 Hertz. So in this study, they looked at both the intra-train correlations between pulses within the same train and inter-train correlations. And one of the interesting aspects, this was done looking at nanoparticle suspensions. And I think one of the more relevant aspects here was that they could follow basically as the sample was warming up during the measurement. So this is now shown in these 2D maps. And this is the same like I was explaining before. Here now, instead of time on the two axes, is the pulse number. So this is the first pulse, second pulse, and so on. And you can see now there is a kind of narrowing that is happening. That means that your dynamics are becoming faster as you're measuring. And by producing a model that takes into account how much of the X-rays are absorbed by the sample, how they're dissipated in the solution, they were able to show that this time here can correspond to a temperature increase. So if there are questions, I will come back to them. I see already some questions in the chat. Or I can also make it now. What do you think, Rich? Should I save it for the end?
I think maybe at the end it would be great if we go through all the questions.
Okay, great. Well, I'm happy that there are questions. Please send me more.
And we have a co-chair, I should mention, Eli Gonzalez, who can read the questions so everybody knows what the questions are.
Okay, that's great. So we'll continue now. I'll tell you a little bit about our samples, our solutions. We looked at antibody protein solutions, and in this case, immunoglobulin, which is this Ig molecule in a polyethylene glycol, and water and ions and so on. And these are samples that our colleagues in Tübingen prepared. It's been a real pleasure working with these guys. I've been learning a lot. Fazung, Zhang, and Anita and Anastasia, they have been the experts in this part of the experiment. And they have been looking at the system for a long time. So one of the interesting aspects in this is that this system exhibits liquid-liquid phase separation. So here is shown a phase diagram, temperature and concentration. And depending now on where you are on this phase diagram, it switches from a one-phase region, a homogeneous one-phase region, into a two-phase region, where you first start having this droplet formation. And this was actually part of the preparation for preparing this very highly concentrated solution, following basically doing a phase separation and taking the concentrated part. And but for this experiment, we did not look at the phase separation. We looked mainly at the one-phase region. And we did that at the MIT station at the European XFEL. Here on the left, you can see Anders Madsen and his team. It's been a real pleasure working with these guys, very nice people. And I should say that the experiment was largely done remotely as well, although we were allowed, some of us were allowed to be there. And on the right here now, you can see how the hatch looks like. So one of the unique features of this hatch is that it has this very long sample to detector distance, as you can see here on the schematic. And this is very important when it comes to this technique, because in this way, it is possible to have enough resolution and momentum space to be able to resolve the speckle patterns, especially if you want to use a larger beam, like we did. So we use the SAX geometry, small angle X-ray scattering geometry, but it is possible also to measure at wide angles. And I should say at this point that a lot of these things in this experiment were designed and driven and analyzed by Mario Reiser, who's also present in the chat. So who is a postdoc in my group, and he's extremely talented, and he has been basically driving this project very hard. You can read a lot more details about the analysis methods in this recent paper we have in collaboration with Felix and Francesco and the other group. So I should also say a lot of this whole project is also initiated by Christian Gut as part of a Röntgen-Angstrom cluster collaboration. And it's been also fantastic working with Christian. I mean, he basically, on the designing part, on the interpretation and everything, we've been working together a lot on this. And that's basically how we came up with these parameters here. I will just show this now for you. I think a lot of people would be interested in that. And I will just point out a few interesting things. We only used 1% of the photons because of issues related to damage and so on. And we also basically, as you can see, generated a lot of data. That was around 800 terabytes from one experiment. And this is a major challenge when it comes to these experiments, basically, and how long it takes from the measurement until the publication. But Mario here has been doing a fantastic work. And this is the layout, like I showed you already. So you have these X-ray pulses, pulse trains that come into your sample. And one interesting thing we could do here, a funny knob that we had, was to tweak the EuXFEL frequency. So it was not possible to go to the fastest, to the highest frequency, 4.5 megahertz, but we could vary at 2.2 and 1.1 megahertz. And this basically translates into this kind of schematic. So that what you would see is that you have by basically changing this pulse spacing, you can distribute the trains differently and therefore probe a longer time at the same. So it's like a way to, if you want to tune your time resolution, and at the same time you can also tune how much dose you expose your sample within a certain time, the dose rate. So the first thing we did was looking at the megahertz SACs. So just azimuthically integrating the intensity. This doesn't take into account the speckles, but just gives you an information about structural changes. And this is shown here now on log scale, the I of Q versus Q for certain dose rate. And now different colors here correspond to different times. So the green one is earlier times, and the red one is later times, which you can also translate it into a certain dose, that is amount of energy that is absorbed by the sample. So you can see here it goes from a few kilogray up to nearly 100 kilogray. And on the inset, we show the intensity normalized to the intensity at time 0. So we see that after 10 kilogray or so, something is growing here. And to quantify the structural changes as a function of time, we plot the Porod invariance, which essentially this is a very common quantity used in SACs to quantify changes in the line shape. And it's an integration within a certain Q range. And here now you see that we observe there is a regime at early times or early, let's say, low doses, which we do not see any major changes. So we call it the non-perturbative. And then after that, there's a lot of things happening. Changes in the form factor, structure factor. We think there's a lot of changes that come after this point. And that is approximately at 10 kilogray. So now the thing that was interesting for us also to look at the correlation functions. And this is this two-time correlation function that was explaining within the train. So here you can see the time constants is in the order of microseconds. And it does show some significant change if you just look at it. It just starts to become more narrow, basically, or it starts to become less pronounced. The contrast starts to decrease. So by taking cuts along this diagonal, you can see, like on the right here, how the G2 function, how the different correlation time changes now as a function of time, or changes as a function of dose, because it's not only that the time is changing, also you're exposing your sample more. And what we see is that there is a significant change in the contrast. It starts to decrease, but at the same time, the line shape changes. So that is something that is happening within a few kilogray already. Now, the next thing we did was vary the dose, like I explained, and they are actually the dose rate. So we can either reduce the intensity, like is shown here. So just by having the same pulse structure, but measuring half the intensity, or change the pulse spacing. And by changing the pulse spacing, essentially, that is another way that it is interesting to understand what is happening within, even though you expose your sample to the same dose, does it matter how fast you do it, how fast you expose it? And that the result is shown here on the right. So you can see here now the correlation function as different for different dose rates. The blue, the orange, this corresponds, and you can see as you increase the dose rate, the dynamics start to become faster, which is in a way not surprising if you think about it. But the way for us to quantify this was to fit it with an exponential. So it's exponential like shown here, where beta is the contrast, the intercept. And this way you can also get the diffusion constant, and alpha is the kWw exponent. So we were able to extract the diffusion constants now as a function of dose. And what we see here is now for the different dose rates is that for all of them, they seem more or less constant up. The diffusion looks more or less constant up to 10 kilogray or so. And then it starts to decrease. So that means that decreasing diffusion means that things are starting to slow down. And that indicates that in this earlier time, we have some kind of increasing D0 as a function of the dose rate that could be related to heating. That's one of the hypotheses. followed by aggregation, which is leads to decrease in the Z. So to investigate this in more detail, we compare our results with the diffusion constants obtained from dynamic light scattering, shown here on the right, at different temperatures. And here, the dynamic light scattering diffusion constants are shown in black as the diamonds, and the EuXFEL data are shown here now for the different dose rates. And we can see for the lowest dose rate, it nearly agrees with the DLS data if you take into account the heating that is induced by the pulses. But when you increase the dose rate, that does not seem to be the case. So there has to be something more than simple heating. The data indicate that there is something more to the story there. Another interesting aspect is we can calculate now the hydrodynamic radius by knowing the diffusion constant and the viscosity. One can get, using the Stokes-Einstein relation, the hydrodynamic radius. So what we saw was that the radius, if you were to normalize the hydrodynamic radius with respect to the time zero, was that in most of, in all of the dose rates, more or less up to 10 microseconds, things do not change. And then suddenly the radius starts to become very large, which is again consistent with some kind of aggregation. And one can plot it in a slightly different way. If you take into account the diffusion constant, so you know basically how far they will diffuse within a certain time, you can also estimate how does the radius change as a function of the root mean square displacement. And we see that all of these different dose rates fall on a master curve if you do that. And that we observe up to 10 nanoseconds that more or less nothing changes, which corresponds roughly to the first solvation cell. And this is the model that we use. So we observe that aggregation sets in approximately after 10 microseconds. In other words, after traveling about 10 nanometers. So the take-home message here is we heard a lot about the diffraction before destruction. Now I want to tell you about this. This is like a way to think about correlation before aggregation. So if you are, let's say, smart enough in designing your experiment and choosing a low dose rates and looking at early times, you can get equilibrium dynamics. At an intermediate regime, where you have some heat, you can have some heating, some driven motion, which we believe also there can be contribution from free radicals also that influence what we observe in the dose rate. And finally, you have aggregation, which occurs for larger doses over 10 kilogray and larger distances over 10 nanometer. And it's a question now whether this is reversible or it has to do with some kind of cluster formation or whether it correlates with denaturation as well. And with this, I'd like to come to the acknowledgement slides. I already thanked a lot of our collaborators and the funding. And of course, thank you for your attention. I'll be happy to answer any questions.